Hello students, this is Dr. Faizan Mirza. Here we are discussing the structural details of our striated muscle. Striated muscle means the skeletal muscle. So I hope you have already gone through the basic description of the three types of muscles. The one which is in detail in our course is the striated muscle, the skeletal muscle. This is how the stripes look at regular intervals when we look at the section of this muscle fiber under a light microscope. And this is at 300 magnification, and you can see individual nuclei here, and you can see these blood vessels being supplying oxygenated blood to the muscle fiber as well. So this is the bone. We know from the bone the skeletal muscle is attached to the tendon, and this tendon allows the muscle to stay attached to the bone. This actually is a connective tissue. So this muscle is actually your skeletal muscle. Now what's this? This is basically a muscle block made up of thousands of muscle fibers. So each of these here is a muscle fiber, and there are thousands of certain such muscle fibers present in this block. And this is completely is one muscle. If you pull one of these muscle fiber out, this is a single strand that you pulled out. This is how it looks like. So this is you are magnifying this to just visualize what this structure looks like. So this is a single muscle fiber. Now in the single muscle fiber, again, you can see there are stripes at regular interval. There is a nucleus present in its periphery, and the membrane of this muscle fiber is called as sarcolemma. So what are striated muscle, type of skeletal muscle? Sarcolemma is the cell surface membrane of the muscle fiber. Sarcoplasm is the cytoplasm of the muscle fiber. Now when we go to the details of the muscle fiber, so this is the short length of muscle fiber here. Again, you can see this is the sarcolemma membrane. This muscle fiber itself is composed of myofibrils. Now what are myofibrils? If we go back to the details of myofibril, we studied that myofibrils are the parallel bundles of contractile proteins, which are present in the skeletal muscle as well as the cardiac muscle. So this myofibril, how it looks like, the myofibril looks like something like this present inside the muscle fiber. So if we take the cut section here and we try to just magnify it, this is what we see. This yellow region here is the sarcolemma. The sarcolemma is the surface membrane of this skeletal muscle fiber. This cylindrical structure is a single myofibril, and this single myofibril appears like a single strand here. So since this is the magnified diagram, so this is your one myofibril. This is another myofibril. This is another myofibril. So there are many myofibrils present inside the muscle. What are myofibrils? Parallel contractile protein bundles. So these are contractile proteins, the dark one, the light one, and you can see they are arranged in parallel bundles. You can see a mitochondria here. This mitochondria will be providing ATP for muscle contraction, and this cytoplasm in between these structures will be called sarcoplasm. Now coming to starting the structural details from sarcolemma. The sarcolemma is the cell surface membrane present on the skeletal muscle fiber. On the sarcolemma, you can see circular holes. These circular holes are called as entrance to the T-tubules. They are not holes actually. They are just T-tubules. It means that the membrane is coming in this direction. The membrane invaginates, it involutes, it goes down, it makes a loop, it comes back again, and it continues on the surface straight. As it continues, again there comes another circular hole, the entrance to the T-tubule. Again the membrane goes inward, it takes a round, it makes a loop, it comes back again. So these are blind-ended ends of the T-tubules, and these individual structures are called T-tubules. So entrance to T-tubule, and this is the T-tubule. Entrance to T-tubule, and this is the T-tubule. So you can see these three T-tubules in this diagrammatic section. In between these two T-tubules, you can see a structure. This organelle is called as SR, or Sarcoplasmic Reticulum. So this is a Sarcoplasmic Reticulum. It is connected by these cisternae to another part. This is also a Sarcoplasmic Reticulum. It is abbreviated as SSR. So the purpose of the Sarcoplasmic Reticulum is to store calcium ions. Now, apart from this, you can see T-tubules I have mentioned, entrance to T-tubule I have discussed, sarcolemma, the mitochondria, the myofibrils as well. So these are covered. In this diagram, you can see again the stripes very well. You can see the nucleus as well. The sarcolemma reticulum can be seen. Myofibrils can be seen. Coming to the structural details of a single myofibril. So this is a myofibril. The myofibril, you can see contractile protein. This is a contractile protein, darker one. This is a lighter contractile protein. What are the contractile proteins? Made up of the darker contractile proteins made up of thick myosin filament. And this one is the thin filament. So the thin filament is made up of protein actin. The thick filament is made up of protein myosin. So if we see the structural details, as I mentioned, this is you can see the arrangement of myosin filaments here. You can see the arrangement of actin filaments. You can see that the myosins are present parallel to one another, the actins are present parallel to one another, and at some point they form an overlap as well. The cylinder is cut at a section where only the myosins are cut. So you see larger spots here. Each spot is actually a myosin. All these are myosin filaments which are cut at a certain angle because the section was taken from here. For example, if I cut it from here, if I take the section from here, I'll be able to see small tiny spots rather than these large spots because they are of actin, and I'm taking the section from here. Now let's suppose if I take the section from here, which means an overlapping region of myosin and actin, I'll be able to see alternate arrangement of one myosin, one actin, one actin, one myosin, and so on and so forth. So I'll be seeing a small spot, a large spot, a small spot, a large spot, a small spot, a large spot, and so on and so forth. Here I'm just seeing the larger spots because each spot represents a single myosin being cut at that end. I hope that's clear. Now, this is the spread out diagram of that same myofibril, and here you can see various labels have been added. Now the label that you need to keep in mind are these are myosins, and the myosins are held together by M line. Similarly, these are actins, and actins are held together by Z line. What's a sarcomere? Sarcomere is the basic contractile unit of a muscle fiber. The distance between two consecutive Z lines is called as the sarcomere. So that's how you define a sarcomere. So one sarcomere is having one Z line, a portion of actin, the whole of myosin, this M line, a portion of actin, and another Z line again. There are certain bands that you have to keep in mind. There's an A band. A band includes the entire length of myosin from one end to another. I band is the region between two adjacent myosins. H band is the distance between two adjacent actins, and you can see that the I band, the A band includes the overlapping region between actin and myosin, whereas the H band doesn't include any overlapping segment between actin and myosin. This is just the segment between two actins, and this just is having the myosins here. So when the muscle is contracting, the sarcomere begins to shorten. When the sarcomere begins to shorten, the Z lines are pulled inwards towards the M line. So this Z line will start to slide inwards towards the M line. This Z line will start to slide inwards towards the M line. So when the sarcomere is shortening, it's shortening in both ways. So this Z line comes as this direction. This Z line will go in that direction, and the sarcomere will shorten. The sarcomere shortens, and that actually helps in muscle contraction. This is the detail here. You can see this is the M line. These are the myosins here, and these individual comma shaped structure or golf stick bob shaped structures, these are myosin heads. These myosin heads are, you can see they are lying at a certain distance from the actin filament. All these myosin heads are the ones which will help the actin to get pulled inwards towards the M line. You can see the orientation of these myosin heads. They are in a different direction here because they are to hold the actin which is above it. The myosins here are pointing downwards because they have to hold the actin from this end, and same you can see the orientation of the myosin heads in this direction. So when the sarcomere is shortening during the muscle contraction, the sarcomere will shorten, and as the sarcomere shortens, how will the bands behave? How will the Z lines behave? So Z lines, as I already mentioned, they start to move inwards towards the M line, and the sarcomere shortens. The M line in the A band, you can see is in the A band, plus the H band, it fills up. So it fills up, and the I band starts to shorten as well when the sarcomere shortens. Now what happens when the muscle is contracting? Here we are discussing sliding filament model. Sliding filament model explains to you how the muscle is contracting and how the sarcomere shortens and what's the purpose of these myosin heads. Here actually you can see the actin and myosin, they are having an overlap. These heads are the myosin heads, and these myosin heads can form a cross bridge with the actin. But this actually is not happening every time. Why? Because this protein which is actin is also having two other proteins in the thin filament. So these rounded structures are all actins. This strand which is running in this direction, this is called tropomyosin, and this protein is called troponin. So all these three proteins together, they make up your thin filament, whereas the thick filament is myosins alone with the heads here. So what actually is happening, the head of the myosin can bond with the actin. So this protein actin is having a binding site for the head of the myosin. The myosin head can just fling up, it can attach to the actin, and it can pull actin inwards. These myosin heads, they work like the wipers present on the car's windscreen, which can move in a certain direction. They come whenever you turn on those wipers, they like start to move one way, and then they go back, and then again they move in that one direction, then they again come back. So the myosin heads move in the same way. So the myosin head can attach to the actin and pull the actin inwards. Now, how is this happening? Why myosin head is not attaching to actin always? Because the binding site present on the actin for the myosin head is covered by troponin and tropomyosin. So when troponin and tropomyosin are in place, they hold the actin in such a way that myosin heads cannot form any bond with the thin filament. In this case, the muscle stays relaxed. Now, what happens? You allow calcium to be released from the sarcoplasmic reticulum. When calcium releases from sarcoplasmic reticulum, it binds to troponin, and the attachment of troponin and calcium here triggers the tropomyosin to alter shape. When tropomyosin alters shape, actin gets exposed, and as the actin gets exposed, the binding site of myosin head on the actin gets exposed. The myosin head, you can see these myosin heads are lying downwards. They will jump and they will hold actin. You can see this actin is held by the myosin head because its binding site got uncovered since the tropomyosin altered the shape. So once this myosin head, it attaches to the actin here, it will start to pull actin inwards. You can see this myosin head, it moves in this direction. First it just jumps up, it attaches to actin, it just pulls the actin inwards towards the M line. So it attaches, it pulls it inwards. So this is the actin going inwards. When it goes inwards, it is moving towards the M line. So this allows the sarcomere to shorten by 10 nanometer. Then the hydrolysis of ATP occur. ATP comes from mitochondria as well as from sarcoplasmic reticulum. The ATP is broken down by the head of the myosin because here the enzyme ATPase is present. It allows ATP to break down to ADP and PI. This causes the energy to be liberated. The release of energy allow the cross bridge to break. This was the cross bridge formation. So you can see that the cross bridge formation is initiated by the release of calcium as it binds to troponin and tropomyosin shape gets altered. Cross bridge formation occurs and cross bridge is then broken down by the hydrolysis of ATP. This is called as the sliding filament model, and this one episode in which the calcium attached and the shape of tropomyosin altered, the myosin head jumped to attach to the actin and pulled the actin inwards towards the M line and ATP hydrolysis allowing the cross bridge to break. This one cycle allow the sarcomere to shorten by 10 nanometers. So you can expect that, for example, if these 5 heads, this 1, 2, 3, 4, 5, 6, 7, these 7 heads of myosin, they attach to actin right now, they will pull actin inwards. So this actin will move somewhere here. So when it is covered this much of distance and the ATP hydrolysis allow the cross bridges to break, since the calcium ion will still be there more, so this actin will keep on moving inwards because the myosin heads are still there to pull it inwards. This is the maximum point to which the actin can get pulled inwards. The actin can never run into the M line because the heads are not present around the M line. This movement of actin along the heads of myosin is similar to singer surfing. Singer surfing is an example that I often give in my class to explain how the actin is sliding inwards towards the M line. You can picture the actin to be the singer in a concert, and these individuals are the people, the crowd in that concert. And when the singer jumps onto the crowd uses their hand and they just allow the singer to surf from one person to another and the singer just is seen moving along the hands of the crowd until the singer reaches the end, probably the end of the auditorium or the arena. And since there is no one beside it, so the singer cannot be handed over back to anyone here. There is just no one to hold the singer. And this is the maximum contraction that can be achieved for sarcomere shortening. So the sliding filament model actually explains to you the shortening of the sarcomere by 10 nanometers. So one contraction, for example, if you want to contract your bicep muscle and you want to just flex your muscle to the maximum, means you are reducing the angle at your elbow to the least. It means you are making a lot of sliding filament model based these sarcomere shortening 10 nanometer each multiple times to allow maximum contraction. One contraction means many times this 10 nanometer shortening must have occurred. This occurring once is called a single power stroke. So you should keep in mind the term of power stroke. Power stroke simply means that since the actin was exposed because the tropomyosin had uncovered it, since calcium had attached to troponin, the actin got exposed and the binding site on the actin for the myosin head was available for the myosin to jump and attach. This initiates the power stroke. So the myosin head jumps, it pulls the actin inwards towards the M line. The ATP hydrolysis then allows us to bond to the cross bridge to break. The head just pulls back in it flings back into original position, and this marks the end of one power stroke. So one power stroke shortened the sarcomere by 10 nanometer. Then since the calcium is still there and the actin is still exposed, this head will jump again, will jump again to pull actin inwards, but at a different site because the actins have moved inwards, and this allows the muscle to keep on shortening until maximum contraction can be achieved. And this all is under your neurogenic control because the release of calcium ion comes from the sarcoplasmic reticulum, and we know that the sarcoplasmic reticulum is released only when the impulse comes from the nervous system. So this you can see here, there are events occurring at the neuromuscular junction, there are events occurring at the end of the muscle fiber. The motor neuron allow the depolarization wave to continue because of the entry of the sodium ions. This motor neuron keeps on depolarizing until this segment is reached. At this point, there are voltage gated calcium channels that allow calcium to enter into the motor neuron. This calcium release triggers the release of this calcium entry triggers the release of these neurotransmitter vesicles of acetylcholine to undergo exocytosis and spill the neurotransmitter into the synaptic cleft. This synaptic cleft here allows the neurotransmitter to diffuse all along and bind to the receptor present on the sarcolemma. This black line represents sarcolemma. On sarcolemma, there will be a receptor for acetylcholine. You can recall that the receptor for acetylcholine is the sodium channel. So when two acetylcholine molecules would have attached to a single receptor, the channel will open up for sodium ions, and sodium ions start to move inwards into the membrane of the muscle. This causes the muscle membrane to undergo depolarization, and as the muscle membrane keeps on depolarizing, the sarcolemma is depolarizing, and the wave of depolarization start to go inwards into the T-tubule. The T-tubule depolarizes as well. The depolarization of the T-tubule triggers the release of calcium ions from the sarcoplasmic reticulum because there are voltage sensitive channels of calcium present on the membrane of the sarcoplasmic reticulum as well. They open up and calcium ions start to move out into the sarcoplasm. This region is the sarcoplasm. You can see the thick filament and the thin filament. When calcium is released in this region, calcium will come and bind to troponin in this region of the thin filaments. The tropomyosin will slide away. It will alter shape. The myosin heads will form cross bridges with the actin and power stroke will occur. The actin will slide inwards towards the M line. This region is the M line you can see here. So this will allow the sarcomere to shorten. Power stroke will occur. The sarcomere will keep on shortening 10 nanometer in each power stroke until maximum contraction can be achieved. What need to be kept in mind is the difference of what calcium ion is allowing to occur. Calcium ion in the sarcoplasmic reticulum when coming out allow the power stroke to continue and power stroke to occur. The calcium ion moving into the motor neuron here triggered the release of the neurotransmitter from the motor neuron. This entire assembly is called as the neuromuscular junction. Neuromuscular junction means that the neuron and the muscle, they are forming a junction. In the neuromuscular junction, basically this is just a synapse, but this is the kind of synapse. In one cell is the presynaptic cell is the motor neuron, the postsynaptic cell is the muscle fiber. The sarcolemma is the membrane at which the acetylcholine receptor is present, and the presynaptic membrane is the region from where the vesicles of neurotransmitter are released. That's it from my side. Thank you so much.